Okay, so today we're going to be picking up with talking about our evolution from early prokaryotic sort of single-celled life and building up to multicellularity, and where we see evidence of that. So there was some of this in the end of Monday, that's the day of the week, Monday slide set. I went ahead and deleted that and moved it all into here so we can have the topics grouped together. So that's just an FYI. Okay. So when we were talking last time, we had seen that we have some evidence of early fossil life in our stromatolites, which are interpreted as probably cyanobacteria in layers similar to things that we see today. We saw those in some Australian fossil deposits. And we also talked about the idea of how we would have gotten from a prokaryote to a eukaryote when we went through a little bit of that endosymbiosis, symbiogenesis on Monday. So now we're going to look through some more of the early fossil evidence. Okay. So other than the stromatolites, I want you to discuss real quick, if you were going to look for a fossil prokaryote, what would you even look for? So other than our stromatolites, you're out looking at rocks, maybe in Australia, maybe in some other datable rocks to the right time period. What kind of evidence might you want to collect? What kind of data? Think for a second to yourself and then talk among yourselves very briefly. About like what tools you would want, what kind of samples you're going to collect. Okay, so what are some of the problems we're going to encounter if we're looking for an early prokaryote? They're really small, right? They're really small. Anybody have any good ideas about how you might look for one? Okay. Well, one, we're starting with looking for rocks of the right age. And I heard someone say microscope, right? We're definitely going to need a microscope because prokaryotes are really small. We're also going to be thinking about some isotope data. So we thought about isotopes and radioactive decay when we talked about dating rocks, but we also briefly mentioned that if you look at ratios of the isotopes of carbon, that there's a specific signature kind of for life. So there's a lighter kind of carbon, essentially, if we're looking at biogenic carbon that was likely part of some type of living creature. Otherwise, we'd end up with our isotopes kind of evenly distributed according to how prevalent they are on Earth, rather than having kind of differential shaded little pockets. So the oldest fossil cells that we think we can see, the stromatolites were nice and big because they made those layers, right? So for those, you can kind of wander around and look for them if you know where they look like. But if we want to look at actual cells, individual cells, we look in a similar region. We're still in Australia. We go to the apex chert. Chert is essentially a type of rock. So this is our fossil location, and this is what they found by sectioning some really, really thin rocks. And they've looked at them in various ways at this point. But one of the more recent things they did, I think, was put it through a scanning electron microscope. So they've been looking through microscopes. They've been looking at the isotopes that they see. They've been trying to see in as detail what they can find, and also like specifically what atoms are there, where are they concentrated, how is the crystal structure throughout the rock. So these microfossils, they are interpreted as fossils, but as we'll see, there's some debate. Date to about 3.47 years old. So we can date these rocks in ways similar to how we dated those zircons when we were trying to find the oldest rocks. We're going to see these through a microscope because they are 10 to 20 micrometers long. And one of the things that they notice about these is that they have these light carbon ratios. So that D is telling you about the ratio of carbon-13 to carbon-12. And we see this lighter concentration of carbon-13 in the chert in like specific areas. So here are some regions that they sampled to look at those carbon ratios here. But if you look at it on a big scale, we have microfossils that look kind of like this. So we have these long kind of structures with some little banding, different shapes here. So when they found these, one of the things that they did, this is an actual data table from a table, they tried to characterize the shape that they were seeing. So for these apex chert microfossils, you can see that they picked out some taxa, so some groups. These are genus, species, demarcations, so an abbreviation letter for the genus and then the species name after it. So they looked at how many they saw, they looked at the widths and gave kind of the range and also the average, as well as how long they were. And then they tried to characterize a little bit about, okay, what's at the end, what's in the middle, and some more kind of shape characteristics here in the end of the table. So this is how they're trying to describe and classify these. Okay. But there has been debate about whether these are genuinely fossils or whether these might just look like fossils, whether they might just look like early cells. So I want you to take a couple minutes and dig through this carefully. So this is from a paper that was rebutting these original findings and saying, hey, wait a second, maybe these are not actually cells. So here's the figure caption, and here is the figure itself. So go ahead and read that and try and figure out which of these you might want to be comparing and what you think about how they look. There's additional data, but this was the piece I think that's most approachable from that. Dim the lights, that might help. So go ahead and talk to each other about what you are looking at, what you're seeing. There are a lot of pictures, so I recommend picking like 2 or 3 that you might want to compare. Okay, so let's start with, let's circle which ones are the microfossils that they're actually looking at. Where can we find those? So we're going to be looking at the ones here that have names, right? So the ones that we're trying to compare are these things in italics, so that those italics tell us this is a genus species name. Holotype, when they first find a fossil and give it a genus species name, a classification, that original fossil is called the type specimen. If it's that goes for living things too. For fossil, it's a holotype. So this is what you compare to if you then find another fossil and you want to figure out whether you need to use a pre-existing name or whether you get to name a new thing. Okay, so let's go ahead and circle those both for me and for you. Okay, so we got C, if I circle the wrong one, let me know. J, M, and Q. Yeah. Okay. So from this description, which are some of the ones that we know are not fossils? That first list, yeah. So here they're saying pseudoseptate and bridge filaments as artifacts. So this artifacts from vein chert, that's a description of like a geological formation. So we can already look at A and B up at the top. And we have our O, U, B, and X. X. And then in this separate section, they're separating out different types of non-fossiliferous things. So here for I, we have a graphite filament. So graphite, kind of like pencil lead. All right, so here we have a graphite filament, EKL. We have some other non-biological structures. Okay. So with those things circled, we can circle some more, I guess, the rest. Could also be one of our fossils. Go ahead and try and find some pairs that suggest to you some interesting comparisons from the biological, essentially, specimens versus the definitely not biological specimens. General, as a table, why don't you take a vote on what you think about these microfossils, whether you think based on this specific data, whether this is more likely a fossil or not a fossil.
One long, I don't want to say one, two, but one, two. And then everything else kind of like this branches coming off. Way over top of it.
Also in here.
So let's take a vote. Who thinks based on the stuff that they're fossils? Got one. Who thinks they're not fossils? Like to see the rest of you raising your hands. Okay, so who doesn't know? Okay. All right. I'm glad I asked for that last one then. All right. So let's call out a couple. I'm just curious which ones you were most interested in comparing when you're trying to make your decision. Anything that either really does look the same or really doesn't look the same?
Like the ones centrally, however, have the structure formed in them, that they like polka dot.
Mhm. So that kind of striping
Yeah.
looks different.
Like recognizing that the, random, branches coming off and more like structures.
Mhm. The
Or
shape
maybe
looks
some-
more
Yeah. Just like
uniform or
under-
something.
Yeah.
And what were you comparing to when you said that?
Like, yeah, like you and you
Mhm.
and Adam. And you
Versus maybe like
versus,
A or something.
Yeah.
Okay.
Or A and B and even the U or not use O.
Mhm.
How they kind of like frame and like flattening. They
Mhm.
don't really have like words.
Mhm. Cool. Does it make sense why people are arguing about this?
Yes.
Okay, cool. So yeah. There's basically still a pretty hot debate about whether they count as cells or not. So one of the things that was also in this paper that was showing these different images was that originally when they were described as being from a beach or a kind of stream. So an environment where you might be more likely to find life, at least as we understand it right now, versus this paper found that they were more likely from a hydrothermal vane system, which we talked about hydrothermal vents before as a potential place where life might have formed. And we do see life near hydrothermal vents now, but the reason that that's suspicious potentially is that you do have all those minerals coming out from the vents, and you might see them collecting in odd ways in that kind of system as well. Okay. I think that the most recent evidence, I think even the people who wrote this paper are now again unsure, primarily because of that light carbon. So that wasn't from the original paper, these carbon and signatures, but looking at the ratios of the carbon isotopes, they did find that the different clusters of things that they think are cells do have lighter carbon than the surrounding rocks, and also that the different species that they've interpreted have slightly different ratios than each other, which might initially make it seem like, okay, maybe they're all random things, but when we're thinking about they're all embedded in rocks around the same plates, if they're formed as part of the crystal structures, you would expect this carbon to kind of be distributed throughout the rock, and you would expect that maybe those little conglomerations to be made of similar stuff, and they don't quite seem to be. So they still go back and forth on this a fair bit. All right. So the next thing that I want us to talk about is looking at oxygen. So as we're thinking about the history of life, when we start, the Earth really doesn't have oxygen in that early atmosphere where we have these early cells forming. So it wasn't supposed to be part of our experiments when we were looking at, do we have amino acid formation from abiotic factors? But nowadays we have about 21% of the atmosphere as oxygen. So that's a huge difference based on what we can see. And oxygen is really necessary for most of the life on Earth today. So it's an important part of the picture. The trouble is, you can't go in the past and directly measure oxygen. It doesn't crystallize in the rocks the way some other things might do, not like in the zircons. You can take ice cores, but you can't really take ancient atmosphere cores. So we have to look at other things when we're trying to find evidence of oxygen. And part of why we're looking for evidence of oxygen, I should say, is that we're thinking about how life works and gets energy. All right. So for the most part, with the exception of some things near those geothermal vents that use that geothermal vent energy for energy production, almost all of life relies on the fact that through photosynthesis, we can go from having carbon dioxide and water in the atmosphere, right, and produce things like glucose, and oxygen is a byproduct of this process. So this is how we store solar energy, store it in things like carbohydrates. And then we can also do the reverse. So if you are a cell, an animal that can't photosynthesize, that can't make your own stored energy in this way, you can take those molecules stored by photosynthesis when you eat a plant, for example, take that glucose, use that oxygen, and essentially run this in the reverse. So you're taking those two things together, you're running it in the reverse to get the energy out as ATP and body. Okay. So this is really how our energy life cycles work right now. So this is why we care about oxygen. So early photosynthesis would have produced oxygen. We assume that this is where the oxygen in the atmosphere came from because we can find a source for it. And until we have oxygen in the atmosphere We can't do aerobic cellular respiration because we don't have available oxygen for our early animals to breathe, really. So we need to figure out how we got that oxygen. Fortunately, while we can't sample 4 billion year old air, oxygen does react with other things. So oxygen reacts with carbon, iron, and sulfur primarily are the things that we look at. So if we interpret or assume as our baseline assumption, okay, we have photosynthesis happening. We don't necessarily have animals and things that breathe oxygen using up that glucose with carbohydrates left yet. So we're going to have essentially leftover oxygen. That's kind of a waste product. Okay. And in any case, in modern life, we still have some leftover oxygen right in our atmosphere. So the easiest one to understand, so the one that we're going to focus on, is what happens with iron. Okay. So in the early Earth, we see a lot of iron. So our current core made of iron, the mantle also lots and lots of iron. So when we were looking at the structure of the Earth, we don't have an iron deficit. We have plenty of it. And when we look at modern rocks and in the ocean, there's plenty of reduced iron and also sulfur. We're not going to talk about it, but iron to sulfur ratios and stuff like that are other things that people look at. The thing to know to understand our next bit of evidence is that some iron is soluble in water and some is not. So specifically, ferrous iron is soluble in water, so it's going to dissolve in the water, but ferric iron isn't. So that is talking about our different oxidation states. So ferric iron is Fe+3, ferrous iron, Fe+2. So that's the difference between those two. So the fact that some of this iron in certain forms dissolves in water and some of it doesn't is going to help us look at oxygen when we look at the reactions we would expect between iron and oxygen. So this brings us to banded iron formations, which are pretty cool. We can actually see these all around Lake Superior. Lake Superior that has a specific named type of banded iron formation. So what happens here is that when iron combines with oxygen, it forms iron oxide. So you have certainly seen that, right? So when your car gets rusty, that's what's happening. And that's why we have paint on the car. So if your paint chips, that's gonna rust over eventually. We definitely saw that on my car recently, right? And we can see that when we're looking at rocks and geochemistry as well. So what do we have happening in terms of our biological systems? So what we're interpreting here is that we're starting with these cyanobacteria. So we have cyanobacteria now evolving in these watery environments in the ocean, and cyanobacteria can photosynthesize. So the cyanobacteria, potentially like those fossils we're seeing from the apex chert, they're photosynthesizing, they're producing oxygen, right? So this creates an oxygen-rich environment in the atmosphere, and we're also going to end up with some oxygen dissolving in the water as well. So what happens then is that oxygen in the water reacts with these ferric iron oxides, okay? Or sorry, reacts to form these ferric iron oxides. Right. And the ferric iron is insoluble. So the ferric iron isn't going to evenly distribute nicely throughout the water. The ferric iron is going to fall down to the bottom. So that is what causes these iron deposits layering kind of on the floor of the ocean, right? So we have oxygen production by the cyanobacteria. We're producing these ferric iron oxides that then essentially precipitate out. So we're turning ferrous iron, which was dissolved in the water, into this ferric iron, these ferric iron oxides. What we then see is that we can, at this point, literally see bands in these old rocks. Right, so this is an example of a banded iron formation or a bit. Before I move forward, notice that these are stripy. I want you to think about what your theories about why we might see this layering would be.
Yeah.
So fun. The reason I'm asking you about the layering is that this is another area where you could dive in and do additional research. So there are different theories about why we see this layering. But I'm going to tell you my favorite one and the one I've seen mentioned most often. Okay. So when we think about these cyanobacteria, right, we are starting out in an oxygen-poor environment. Our cyanobacteria, they're photosynthesizing. So I heard someone say something about changing environment. Right? When we think about changing environment, that can happen on multiple time scales, right? So we live in Duluth. When you think about the changing environment in your life, what would you be thinking about? Seasons, yeah. So one of the ideas about why this layering is occurring is that this is showing something like seasonal conditions. So you can kind of relate this potentially to what happens in a lake or in an ocean. Sometimes they send out warnings saying there are really high bacterial levels, there's an algae bloom, don't go in the water, right? And that happens somewhat seasonally. So if you think about cold environment, right, we'd expect all sorts of processes to be slower, right? Because among other things, just chemical reactions happen slower in colder environments. So we might expect things to go sort of dormant, either during something like a winter season, or if there are a series of years that are colder than others. And then we might also expect that there are times of year or actual chunks of years where the conditions for the cyanobacteria are really nice. We have a boom in population potentially. And during this boom in population in, say, maybe the spring, the summer, or a chunk of really good years for some other reason for the cyanobacteria, we end up with sharp rises in literal oxygen levels during those time periods, at which point we end up with a lot of iron deposits. Then they die off, maybe go dormant, maybe just there aren't as many of them. We have other layers of sand and stuff depositing sediment on top until we have maybe another boom. So that's my favorite of the ideas about how and why we might see these different ones. Did anybody have any other ideas that they really like? A lot of silence at the beginning, so it's okay. You don't have other great ideas, but I like that one. That's one of the ideas. So if we look at the presence of these bits, these banded iron formations, we can also see that they're not consistently present over time. So when we're looking at our time scale, timeline, so here we see the present day at 0. Here we see 4 billion years ago, so near the beginning of the Earth. Right. So what we see is that for a couple billion years, so 2 or 3, we really don't have these banded iron formations. So we don't have this signature of there being a bunch of oxygen in the atmosphere. So then we have a super obvious surge in banded iron formations, and we can see them across the world. So we can see these banded iron formations in Africa, Australia, Eurasia, North America, South America. They're kind of all over the place. We can see that we have them in a similar sort of time scale, right? So we're kind of clustering around about 2 billion years ago. Remember the GA billion years ago. Then we see them slow down their formation and actually disappear for chunks of time. So thinking about oxygen and how this formation is happening, why might these stop forming? So you can see they're not forming as far as we know today. So you could start there. Why do you think that we're not forming these banded iron formations today? Definitely lower than back then, for sure. Yeah. We do have high oxygen levels. Yeah. We also have consistent oxygen levels, right? So there was also this banding structure, right? So that's one of the pieces of how they form, what they look like. Okay, so we would expect that as the oxygen levels are changing, in this case, rising, right, that we're looking at this layering as we see a change, and potentially that on some sort of a time scale, we see surges of oxygen and then drops of oxygen. In modern times, we have a pretty consistent oxygen level in the environment. So we have a pretty sort of consistent kind of turnover going on when we're thinking about oxygen and iron oxide formation. So we do have some iron coming up through plate tectonics and crust and a few volcanoes that we do have, but we also have lots and lots of plants and animals all around, kind of stabilizing these oxygen levels at their current spots. So that's the idea about that. So that's also part of why we interpret these as records of oxygenation of the earth kind of starting, right? Because if they were still forming all the time now, then we might have like kind of more questions about that. In addition to the fact that oxygen is just important for like us to breathe, so we really care about it, there are more implications for what oxygen is doing in the early Earth and how it's creating kind of a current condition. So before we have oxygen in the atmosphere, we had a lot of methane gas. So you can tell me something about methane. Yeah, it smells. So methane smells, and it's also a greenhouse gas, right? So I'm sure you've probably talked about that in global, right? Okay. Sometimes people say, like a soundbite I like is like cow farts are a big cause of global warming, because I think that's very funny, just to the smelly bit. And it also helps sort of relate to this first piece. So as we have oxygen kind of replacing methane that is in the atmosphere, we end up with some cooling. So there's evidence that early in the Earth's history, there was actually kind of froze over. So we have one of our earliest ice ages in the deep past. And one of the ideas about this is that this is as the atmosphere is shifting, less of the methane, more of the oxygen, so less of this kind of warming effect creates that disruption. The oxygen also helps create our ozone layer. So oxygen literally reacts with the UV rays from sunlight to form ozone. So as this is happening, creating an ozone layer, which we have today. The final thing we want to think about is, we call this the great oxidation. Did I mean oxidation or did I mean oxygenation event? Both, regardless. So as we are creating this oxygen-rich environment, we said that we have cyanobacteria. We're looking for other evidence of life in the past. Do we know anything about oxygen that we might want to think about when we're thinking about that life existing? So I'm going to tell you that for things that don't breathe oxygen, oxygen is kind of poisonous. Oxygen is reactive. So one of the things that we interpret as happening around the same time is that for things that weren't making the oxygen, we end up with not one of our named extinctions, but we suspect that this would have created a big dying off of a lot of the early prokaryotes, a lot of the early cellular life that hadn't developed photosynthesis and the ability to kind of tolerate oxygen. So as we walk through time, we're going to be thinking about how major shifts in the environment cause major shifts in the life that is present on Earth. So we can see that better when we can physically see the record of the life, see it better as it's closer to the present. But we do think that this would have happened quite early on as well. So those are our early fossils, and that's what's happening as we're moving into our eukaryotes that have organelles like chloroplasts. So we have photosynthesis. And our next step is we kind of want to move towards multicellularity because we want to think about the cells. We want to think about things like animals. Okay? So as we're moving towards multicellularity, I'm going to make you talk to each other a lot today. Why don't you discuss briefly with each other why multicellularity is advantageous? Why is it helpful? Why is it good? Why might you want to be a multicellular organism?
I mean, you're done. Yeah. No.
So what are a couple factors we're thinking about when we're thinking about multicellular organisms? What do we think some advantages would be? Better than one, longer life. Okay, cool. Yeah. So we have sort of backup systems so that we can live longer. We're working together as a cohesive community so we can have specialization, so we can end up with basically more functions that we can perform if we have different organelles in different cells able to do different things. And another thing about multicellularity is that we can physically get bigger. There is a limit on how big a cell can be. So if you want to be something big on top of the food chain, it is also advantageous to be multicellular. Okay. And the reason for that is that cells really get their nutrients through diffusion, essentially. So you've probably seen something like this in your early bio classes, right? But certain things can get across the cell membrane, certain things can't. So if you are a little bitty cell, you're getting things like gases just dissolving directly across your cell membrane. It's helpful, right? You get your oxygen to breathe, you can breathe out your CO2, awesome. But when we're thinking about diffusion works essentially by just kind of like waiting until something hits your surface and then moves through. And then you also have to wait for stuff to kind of like diffuse around on the inside through the cytoplasm potentially. Like there's time scales associated there. So cell size is actually constrained by the surface area to volume ratio. So you can think about it kind of like a pizza, right? So if you were waiting for the inside of a tiny cell to get an oxygen molecule, right? You're just waiting for oxygen to bump into the cell layer and come in, right? Maybe it's being carried around by hemoglobin. Maybe you have maintained specific oxygen levels in your blood or something, but fundamentally it still has to come into contact with the cell. Right? It doesn't take all that long for it to get wherever it needs to go. So that happens when you have a relatively large surface area relative to the center. But if you think about a really big cell, you're still waiting for example, oxygen to bump into the outside. But it's going to have a lot harder time kind of like dissolving and diffusing across the inside and getting where it needs to go as you get really, really big, because you're limiting kind of that outer perimeter and increasing the volume. So at a certain point, this becomes just like a bad way to do stuff. So the cell can't get too large and still easily be able to get really anything moving through by diffusion on a useful and consistent scale to the center. So if we want to get big, we have to get multicellular in addition to allowing us to specialize. If we're big, we can eat smaller stuff. Better to be the big fish until there's someone fishing. Right. So when we think about early multicellular life, we do think that it started with communities of unicellular life. So one of those communities we saw in the stromatolites, right? We see these biofilms kind of forming. And from genetic evidence, we kind of think that one of the earliest forms of multicellular life might have been something like a choanoflagellate. So this is a protist. And when we look at modern sponges, we can see that they have cells within them that actually look very, very similar to these choanoflagellate protists. So these sponges kind of look like giant communities of protists, but these have become specific choanocyte cells within the larger sponge, which sponges actually are animals. So sponges are multicellular organisms, they are animals, and they have structures in them that look like these cells we see other places. So when we trace back genetics of these structures, we also find a really, really far back time for the split. So we think that possibly this is one of the things that was kind of happening. In terms of what we see in the fossil record, once we're multicellular, we can get bigger, which means we can start to physically see more fossils. And they're easier to find because we can just kind of straight up see some of them instead of hoping that we section the exact right rock to find a tiny little microfossil that we can then find the carbon isotope signature in. So at 2.1 billion years, we have something called Britannia spiralis. So for the spiralis, right, we can see this kind of spiraling structure here. Right, so here you see it in kind of an animal interpretation. Here you see it on a fossil cast. And here you see it like in sediment. Okay. So our current best guess is that this is a eukaryote and that this is evolving multicellularity. It's really nice that this is macroscopic and we can find it. Okay, so this we very, very solidly think for sure is a fossil, right? We don't have to argue about whether it's a cell fossil or just some random chunk of something. This is pretty clear that it has organization, easier to find, we can study it better. It's also cool that you can find them in the UP. That's where they found them in Michigan. Okay, so as I said, rocks around here are really old. And as we move a bit forward in time, once we're already just to 2 billion years old, we are now looking at a time period where we could potentially go somewhere vaguely nearby and start to find fossils. So we interpret them as eukaryotes and multicellular because of this complex structure, because of their size, essentially. We're looking for other early evidence of fossil multicellularity. We can look at a microfossil from Scotland. I think there have also been some recent good ones from China, not necessarily this exact thing, but around similar time. So this Scottish microfossil is about 1 billion years old, and we can see it over here. So these are their microscope slides, essentially. And this is their graphical abstract. love the idea of a graphical abstract for scientific papers, by the way. Now some journals ask for them, and I think that's an awesome idea. So this is a new genus that they named Bicellum. This specifically is Bicellum braseri. So what they think they're seeing here in this microfossil is they think they're starting to see cell-to-cell adhesion. That's why we think that this is showing early evidence of multicellularity. The one thing is that they're noticing that they have two distinct cell types in this fossil, right? And they think they actually see a life cycle as well. So they think what they're looking at is that they're starting with a naked stage here that over time differentiates and the cell gets longer. So they start to see their two different cell types. And then as it continues through time, they end up with a cis stage where those two different cell types separate into layers, with one of them creating the outer layer. This is a direct quote from their article where they were reporting this. They were talking about this mature form of their fossil organism, describing it as a sphere with tightly packed cells enclosed in longer kind of sausage shaped cells. So maybe that can be seen best in E, I would guess. I didn't copy the caption here for you. So we can see that we have a long sausage shaped cell here on the outside. And we can see more tightly packed cells here on the inside. So they're interpreting this as growth with cells that are clearly adhered to each other, working as a single organism. Okay. Okay, so this should be doable. So what I want you to do before we move forward into our next unit for Friday, which I guess we'll just do on Friday. Perfect, because I wasn't sure what we would get to or what we wouldn't. I want you to go ahead and start reviewing the chunks of the geologic timescale that we have done so far. So specifically, I want you to go consult that geologic timescale and make sure that you can redraw the Precambrian. Make sure you know what the Precambrian is, what the eons are, what the eras are. And I want you to go through our slides, and for the things that have years, I want you to mark them on this little drawing. So I want you to mark where our really old rocks are, and where the couple of fossils that we picked out as evidence would land within that kind of time scale. We will quickly be moving into an eon where we can start to see. That is it for today, and we have 5 more minutes. I bet you can do this in 5 minutes. So if everybody has a piece of paper, do it now. If not, you can bring it for next time for homework.
Tomorrow. Okay.
That'll be fine.
Okay.
Oh, and let me turn this off.